Okay, I give you 2 minutes and then I go to the Menti doc. Yes. All done. Can I skip the slide? Okay, so I see critical thinking, IAs, research, personalized platform, collecting data through experiments. So when I see research, personalized platform, yes, good. It is something which is connected to the person who is conducting this IA, who is taking care of this IA. But there is a twist now with the new curriculum, which has been first assessed in the year 2025. It can be an individual investigation. Along with it can also be a collaborative work amongst the learners in the same class. So coming to the next point, collecting data through experiments. Data has prime importance in IA. Not only data collection, but also the analytical aspect of data analysis. So yeah, good. And critical thinking, of course, critical thinking is required if you want to pursue any scientific investigation. Without critical and creative thinking, it won't be a very successful one. So coming to today's agenda, there are 6 important sections where I want to just share our thoughts, share my thoughts with you and what you feel about it. So we will go one by one. There may not be like a very clear demarcation when I'm speaking, but we will go one at a time so that it's easier to follow, at least for the learners who have joined today. So just giving a brief overview, what is the biology IA? It is an integral part of both SL and HL in biology. Now, a very important note which I want to start this webinar with is, it is indifferently to be pursued by the standard level and additional higher level students. The criteria rubric, the marks, the scores, all remain the same for IAs. So it is never a thought to be given of that I am an HL student, so I have to do a very complex investigation. No, it is not like that. Coming to students' original and authentic work, many times due to my experience, I know students come up and they are very stressed to find out an investigation which is like very new, novel, innovative. Dear students, it's not a discovery. You won't be given a Nobel Prize for it. Authentic and original, no need to stress. It just means that the data collected, the method developed or the method practiced for data collection, analysis and evaluation is your own work. It need not be a novel work. So one suggestion which I have also included in this is, always use a topic which is of interest to you. Because though the time limit given is approximately, it should be at least 10 hours, which is allocated in the curriculum, our biology IA at least is not possible to be completed in 10 hours. Whether you do a growth experiment, you do an enzyme experiment, according to the magnitude of data which IB has prescribed. So IB says 5 levels of of independent variable which you have selected, and at least 5 trials for each level. So here you get 25 values or 25 data points, and you have to also plan a control for your experiment. Remember, none of the scientific experiments are valid without a control, because we don't know what could have changed in the experiment which gives you those results. So for the validity, you need another point, which is the control. When it's a constant point of doubt, every student asks, is control included within those 5 levels? It is not. It's an extra point. And so in this way, you have 30 data points. Your data size is 30 samples. So it is not possible that if you are not interested in the topic, you don't want to dig deep into it to complete it or accomplish this. Okay. 20% contribution to the final IB grade. This is also a very important aspect of IA with respect to the learners, because 20% contribution, 1/5 of your grade, is totally in your hands. You know, the other 80%, which is because of the IB exams, it is very unpredictable. You don't know the day on which the exam is there, what will be your status, what will be your mood, how much you will be prepared, whether you will be in a better state of mind to write the answers or answer the exams. It's totally unpredictable. But this, with the guidance of your teacher, is perfect, a perfect situation. IA is not for punishing or penalizing the student. It is not that the teacher cannot help. The teacher can mentor, counsel, and guide. Even the student chooses his or her topic of interest, and he or she can take the help of the teacher, or should take the help of the teacher, to understand the feasibility of the research question. Feasibility means you choose a very good aha topic, and then you find out whether you have the resources or not. You plan everything, you write everything, you dig the research, review, literature, everything is done. You have already completed 5 days from when you have started. And then you come to know, okay, this cannot be done because the necessary equipment is not there. So right from the beginning, your teacher must be informed what you are doing. Again, as I told you, it may be individual or collaborative, but the RQs must be different. So collaborative, there is a big doubt nowadays. What is collaborative? IB says the RQ needs to be different. The data collected needs to be different. So I will give a very quick example. Suppose it's a very favorite topic, effect of temperature on enzymes. So, okay. Effect of temperature on enzymes, you can do the planning together. You can take the glasswares, you can make enzyme solution, you can do the research review of literature together. But your RQ needs to be different. So that means your source of the enzyme can be different. Your enzyme can be different. One can choose the protease enzyme. One can choose an enzyme which is like amylase, or the factor is different, which you are choosing as the IV. Either you chose as temperature, pH, salt concentration. So it must not be an exact copy of what the other person is doing. But during the method, one student can help other with their insights. Suppose you want to prepare the same buffer for the enzyme solution. It's a collaboration. Once the buffer can be prepared by you, the next time the buffer can be prepared by your peer. Okay, but the RQ, the data collection, needs to be different. Develops research and analysis skills. Yes, it's also like IB is a holistic education. So it not only focuses on conceptual knowledge, content knowledge, but also development of skills. So it's a right inclusion for the IB curriculum. Now coming to IA investigation, being a teacher myself, I always suggest my students to take a wet lab experimentation investigation. Simulations are available and they are accepted. The use of simulations are accepted by IB for your IA. Extraction, analysis of data from a database, and that too is possible. Field work is possible for mostly the ecology IAs. Use of spreadsheet for analysis and modeling is possible. But being Group 4 science, experimental sciences, I recommend you for high scoring IAs, please do hands-on practical experiment in the laboratory. That is always a recommended one, but no one can fail a student or give bad grades if practical work is not done, and if the student has analyzed the data, collected the data using a simulation. So that's it. We all know that now there are 4 particular criteria in the assessment rubric for IA. I will not go over it in detail because it has research design, data analysis, conclusion, and evaluation. None of them are more important. None of them are least important. They all have been given equal weightage, each weighing 6 scores, like scores of 6 marks, totaling about 24 for the whole IA. Okay, so I'm not going through the assessment rubric or the criteria. We will, if there are any questions, I will cater to them at the end. We will quickly go for the benefit of the students to the actual IAs, how we go on. Okay, so just to have a better purview, please scan the QR code and vote for the common myths. There are many myths regarding IAs. Please scan the QR code and you will be directed to a poll where you can vote for the common myths. The myths are also listed in the poll. And after you vote, we will debunk those common myths together. It's in Quizmaker. So the first question I see is, the more complicated the experiment, the higher the marks. True or false? The second myth is the IA must be entirely unique. And the last one, I need sophisticated equipment or complex methods to score high. Okay, now we will move to debunk these together. Let's check. Yeah, so we are here. The first myth, more complicated experiment, higher marks. Complexity does not equal marks. Clear scientific thinking does. Clarity in your scientific thinking can be very much evident by the analysis, what you are presenting after you collect the data. The IA must be entirely unique. As I told you earlier, it needs to be authentic work, not a Nobel Prize winning discovery. I need sophisticated equipment or complex methods. Even simple tools used with high precision and simple designs with deeper biological analysis score better. Okay, so these are just myths. There are no facts on this. Now, this is, according to my experience, this is actually the idea matrix from where mostly the students chose the topics. Biochemistry, vitamin C, photosynthesis, enzymes, respiration rates. Similarly, few topics are very common from ecology and plant biology, physiology and microbiology. But you have to keep some points as reminders. First of all, before you choose the topic, please go through the science experimentation guidelines. If you don't find one on internet, please ask your teachers. They can provide you the guidelines. So IB has very discreetly framed these guidelines. Many a time students come up with the same IA, copying the same RQ. Not recommended at all. Because you will not have any scope of your independent thinking. So you can take a guidance from that which type of IA topics can be taken, how the IA topics can be can be modified to suit your interest, suit the like when you assess the feasibility, what equipment is there in your school laboratory, what resources are available. You can also choose the IA based on your interest and also the expertise of your teacher, so that the teacher can guide you well. So there are many wise decisions which you have to make. Similarly, many a time students come up that I will come up with the experiment on heart rate variability or color blindness of the subjects. Remember, all these subjects, I mean to say those who are your experiment subjects. So these experimental subjects, you need their consent. There is again there are many challenges which you can find in those, because just taking one subject will not help you. You need to take a particular age group, gender, health condition. There are many predisposing factors before choosing the subjects. And we all totally agree that the IB students are very busy throughout these 2 years. So a bit of extra work may be penalizing. So choose with a very solid mind what you want to do. Make some smart decisions. So here, if you have a broad idea that I want to work on this topic, you have this funnel that can a research question be formulated from this topic? Can it be verified? Can it be investigated? What are the variables which I will take or select if I take this topic? If I want to work on any organism, that plant seeds are very convenient to work with. Whenever you come to work with some animal life, it is a total no from IB. Also, if you choose microorganisms, because there is a lot of fuss with microbiology, biotechnology, being a microbiologist myself, I am very inclined that my students work on microorganisms. But you can surely work, because that has some future, lot of future scope. But you have to be very careful. Read the science experimentation guidelines given by IB. A handful, I can just tell you. Like, for example, if you want to work on any bacteria, your school don't have the permission, none of the schools have the permission from IB to store or to culture these bacteria. Your teacher needs to place an order from a recognized culture collection center with proper documentation following the procedures, that you have a biosafety level, like without any biosafety considerations and organism which can be handled at your school. Even the culture collections ensure that whether you have a laminar airflow and autoclave in the school, these are the minimum requirements to work with any microorganism in the school. Next, sorry. Next, you must not culture or grow any of these microorganisms, because being bacteria, they have the optimum temperature of growth at 37 degrees. You should not culture any of these microorganisms beyond 30 degrees, because then they can also be opportunistic pathogens based on the immunity levels of the people using your lab. Okay, so there are many considerations which are experimentation considerations, ethical considerations, feasibility considerations. Based on all that, you have to choose a topic and formulate the research question. Also, the research question, when you formulate a research question, your research question should be like, should be answerable. It should result into an investigation, and it must be answerable using a biological concept which is in your curriculum. IA and EE, extended essay, there is the difference. EE is beyond your curriculum. IA is within your curriculum. Okay?
Ma'am, can I ask one thing?
Yeah, sure.
Yeah. Hi, Deepa ma'am. This is Nidhi. Ma'am, can we have secondary research as well for biology?
As I told you, yeah, secondary research, database, many things are possible.
Hmm.
Many a times students don't analyze the feasibility of their IA and they don't do a pilot trial. They start directly doing an investigation. Many a times their investigation fails or there are no concrete. Now, when I say investigations fail, it does not mean negative results. It is that none
Hmm.
of the results are coming. So, for example, when a student does a seed germination experiment,
Okay.
so we need to be very mindful that the appropriate type of soil, if
Hmm.
the plant health is studied, so the appropriate type of soil, the season when the seed will germinate, that particular seed needs to be taken into consideration. Now, if the experiment has not no scope in that season or in that environment, then it is not a negative results. It's a bad choice of experiment because we
Okay.
haven't done the pilot trial. At that time, I normally recommend, if there is a case, you use secondary data. Otherwise, there are
Hmm.
many subjects like psychology where only secondary data is taken.
Yes, but ma'am,
Uh,
like for if I want to study pollution effects and all those effects at that point of time, do you think because a lot of data we can take it from is from the websites, right? So there we can use them, but how will it be seen as a poor IEA in IB, or we can ask students to look at that also?
I don't recommend, I don't prefer. Then what is the difference being biology, physics, chemistry in the experimental sciences? Group
Yeah,
4
that's
is
why. Yes.
experimental
So
sciences.
one of my kid is like, I don't want to do touchy touchy. What can I do? So I have to convince her to be touchy touchy.
Yeah, just it's not necessary that they do some very extravagant work. Even seed germination using different salt concentrations can get
Yes,
done.
I believe in that only, but she wanted something just completely. She didn't want to do anything in bioinfo. She didn't want to do anything on the so and anywhere she has to touch the sample even with the gloves. So I was struggling. So I thought I'll ask you this question. But anyhow, thank you so
A
much.
driving force for the students is we should not, and the students are here, they may feel that such a cunning teacher is Miss Deepa, but I often say, if they are like that, I said, first you have taken biology, you have to do something which is touchy, or you get a 4. Are you satisfied with a 4?
True,
Then it's
very
fine.
true.
Then it's fine. Okay. So I hope I answered your question, Nidhi.
Yes, yes, ma'am, yes.
Yeah. Okay. So here is a chat challenge for all of you. I have 3 RQs on the screen, and just in the chat challenge, put in order which one is the strong one, which one is the moderate one, which one is the weak one. Just write on in the chats. I don't see. In the meantime, I'm seeing MD Deepthi, right? In the beginning, someone said.
Right.
Yeah.
Yeah. It's deep.
So, Deepthi, a student can take standard syllabus experiment and make it unique enough to score high. Yeah, of course. Just they don't follow the regular method, but they develop something, a method which can be very much indigenous, very raw. It will add to a high score.
Sorry, ma'am, can you repeat Deepika's question and the answer again?
The question is, if a student takes a standard syllabus experiment and make it unique enough to score high.
Okay, all right.
Yeah. So some additional from the normal curriculum or the regular experiments. It can help.
Yeah, thank you, Deepa, because many a times we see that most of the IAs they are based on the regular topics. Like if I say that the same topics are being repeated again and again, so just taking the same topic, simplifying it, and uh, presenting it again and again, it is not putting a good impact. So what if we just explore the different ways the same topic can be described and it can be researched upon, so it can also help a student in exploring the various aspects of a single topic.
Yeah, yeah. That's okay. That is doable.
Yeah, thank you, Deepa, for your answer.
Welcome, welcome. Okay. So we move to our slide. Okay, so here it is. So what is the effect of music on plant growth? It's a very weak RQ. It's very generic. Which type of music? What will be the frequency? Plant growth, which plant? How the growth will be measured? Then comes the moderate one, where they have given how does the variation in light intensity. So at least we know that there will be different levels of light intensity and affect photosynthesis in aquatic plants. So the plant growth can be assessed as photosynthesis, and the plant will be an aquatic plant, not a generic plant at least. But a strong one says an investigation into the effect of 100 ppm aspirin on the root length of the bean plant, which is Phaseolus. So that is a strong RQ based on these 3 questions, but we can make it more strong if we write the scientific name using the proper guidelines for writing scientific nomenclature. Also, how we will measure the root length, if we can make that as we can consider that in our RQ, that will be also helpful. I don't know. Yeah, just Can you all see my screen?
It is
Hello?
appearing gray.
It's appearing gray.
No, it's gray.
Okay, let me reshare it. Is it now okay?
Yes.
Okay, we'll just put the slideshow. Yeah. Okay. So coming to for the students, particularly this slide, research question framework. So there is a particular framework for formulating the research question. What all you need to include: independent variable, dependent variable, control variables. To be little faster, I will add here only control variables for a high scoring IA needs to be at least 5. According to a scientific investigator, you should not miss any control variables, because if you miss any control variables, your results, your data is not valid. But still, for IA, knowing you are a student, at least 5 is a must. The RQ needs to be an open-ended question, so you know it should ask, it should start with how, or to what extent, um, enabling it to be reasoned, to be analyzed. You should also include the study system or the method. For example, you should include the which enzyme you are using, which from which source, or if you are having a very famous method which you are using here, maybe like the um, bubble like bubble movement method for collection of oxygen, any method, or you are doing protein estimation, Lowry's method. So any standard method, if you are using in your IA, you for measuring the DV, the dependent variable, you can also include that. If you mention any organism in your biology IA, ensure you write the scientific name of that organism following proper guidelines. Now we will just see a research
When you
question.
say I have to mention the name of the organism following a guideline, you mean only the organisms we will pick which are in the guideline, right?
No, 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 no. There
Then
are no organisms name given in the guidelines.
exactly, that's what confusing
You
me.
have to write the name of the organism following the guidelines for writing scientific nomenclature.
Acha, okay, sorry. All
Yeah,
right.
yeah. So here is a simple question. How does temperature affect enzymes? What type of question quality do you all feel? Very good, excellent, poor, average?
Poor.
Poor. So how we can evolve? So this is the raw idea, how we can evolve this question. This is very generic. They have given temperature, we don't know which range of temperature, we don't know which enzyme. So we will evolve it to a better version where we say, like, how does temperature affect catalase activity? If you follow the framework given for RQ, everything you can include and you can make it an excellent RQ. For example, how does increasing temperature? That means I'm changing the temperature. It is the IV. The level of IV is given 20 to 50 at 10 degrees centigrade intervals. Affect the rate of catalase activity. So I know the DV is the rate, the source of catalase is given, and the time duration is given. You can include control variables in your RQ. This is a very frequent question from the learners. Can we include control variables? Yes, you can. So, for example, if we have a question, the effect of music on plant growth. For example, the poor question, which we reviewed earlier, it's very vague, untestable. Nothing is given. No plant, which plant it is. It's a tree, it's a plant, it's an aquatic plant, it's a herb. We don't know. Effect of music, which type of music? So a better makeover for this question can be, it's just to give you some examples. What is the effect of various frequencies of sound waves? We give hertz. It can be made even better by adding the frequency range and the interval. On the germination rate, if you add the unit of germination rate, so the germination rate can be per hour, per rate is always measured. Now there is always a confusion between germination rate and percentage. So provide the appropriate unit of Raphanus sativus. This is the way you write the scientific name. Seeds over 72 hours. So 72 hours is the control variable. The seed name is a control variable. We will take seeds of Raphanus sativus only and not Magnifera indica. So this is how you can make over your RQs. It is not necessary that in the first try you give a full, complete, excellent RQ. You can improvise your RQ with the help of your investigation, with more and more exploration. We will just move from this, so that it's just a situation. And I have asked to identify the flaws, but I think we are running short of time. I will just quickly go to the experimental design. So you see the experimental design. When you see, I told you repeatedly, IV needs to be identified, DV, control variables, at least 5, materials and method with a control, and repeats. Control and trials are must. Here is a variable mapping, the missing link, which I thought of to spot the error. So the scenario given is seeds placed in trays with different soil pH levels. Germination rates recorded over 10 days. So you see, what is the IV? It is the soil pH. We can take a range of pH using buffers. DV, it is germination rate. Now, can you spot some controlled variables in the chat? Please add some control variables in the chat. I will move in the meantime. So these are the control variables. I have identified 5 of them. Volume of water, temperature, volume of water for watering every day, or the frequency you can mention, temperature, light exposure, seed viability, impact of pH on soil microbial activity. So all these are control variables because we can choose the same soil, place all the trays in the same ambient environmental conditions. Just consider, if you miss any one of them, will your investigation be a fair one? So for the scientific investigation to be fair, it is essential choose or identify as many control variables as possible. Many a times I face this challenge with my students. I believe all the teachers might be facing the challenge. The students are not ready, or they don't find enough time to conduct a pilot trial. But believe me, students, a pilot trial saves you from many other different hurdles for your IA. So, for example, you start with very broad values. For example, suppose here one example is given. I will just code a more relevant or connected one, which may be good for you. So, for example, you are choosing the temperature range from 10 to 50 degrees. Now you are choosing for the enzyme temperature of 10 to 50. If you choose at the interval of 10 for the pilot trial, very good. You just do one trial with 50 degrees. The enzyme dies. The enzyme is killed. You are not getting your results. So what you say, this enzyme is not working. Come to the teacher, say this enzyme is not working. It is from an old bottle. No, please make some intervals, some levels, so that you know it may be not active at 50, but it may be active at 10. So pilot trial with broad intervals are essential, and it saves you from many other forthcoming challenges. So instead of being a dead end, just try to work on it so that you can save your IA. So you see here, I have
So,
given
ma'am,
one.
here, sorry, Deepa, ma'am, to interrupt. But when you say pilot study, so
Yeah,
let's say I'm doing a temperature one only. So kid have
yeah.
picked up five temperature. Now, when they are running at those five temperature, if they're checking one or two temperature, so wo data bhi to they can use later on, right?
They can use, but according to the validity of your experiment, your whole trial needs to be done at the same time or with the same conditions.
Yeah. So I'm saying if they're getting result, then why don't they conduct it for all?
I didn't get you. It also specify which topic you are talking about.
If for enzyme only, let's say they get a result at 37, okay,
Yeah.
then they do it and they got a result for 40 as well, right?
Okay.
Then if they're getting result, it means it's right. Then they can there and then, instead of doing it again, there and then they can
Yeah.
take it na, hai
If
na?
if the student chooses that
Hmm.
he or she will take the same temperature range and it's working, each one is having a significantly different result, it can be taken.
It can be taken. Now
Yeah.
for plant
But
germination
remember,
thing, let's
in the
say. Hmm.
pilot trial, often I have seen the students don't take the repeats. They just do one
Yeah,
experiment.
because they treat it as pilot only, na. So they just want
Yeah.
to check if things are working, if the
Yeah.
method is
So
working or not. So they are
so then
never prepared at that point to carry forward the things.
I understand. I'm not blaming. I'm just saying that then for the 5 trials which they have to take, the 4 trials may end up with different values than this one. Because at this time they were not properly trained. This was the very first elementary one.
Okay, so it's kind of a thing. Let them run, see how it works, and when
Yeah.
they have all their flaws, work on it, and then go back
Yeah,
and redo it again.
there can be some adjustments, like no need to take all the 5 temperatures in the pilot trial. Take only 3, so it's quicker.
Okay, okay.
Yeah. So when you do without a pilot, you do execute the full experiment and then you discover later that it was not working. Then the data becomes unusable. The original plan with the pilot saves you from these concerns and you have a better scope for your IA. So that is the conclusion what I can say for your pilot. Safety and ethics, often the students neglect. They don't write about it, but it is also given in the assessment criteria. So please give the risk which are involved in your experiment, what precautions you have taken to avoid those risks, and how it can impact the ethical considerations or the environment. It is a must and it is a mandatory thing. Coming to the data synergy matrix, often the students say that the qualitative data is irrelevant or it is not necessary. Biology is a science of observation. Qualitative must supplement the quantitative one, because if you collect the quantitative, you have the data, you have the figures, you have the numbers, values, but to connect it to a biological reasoning, it is very essential to have the qualitative data. You observe, you connect with the biological context, you enhance, you can improve, you can improvise your method using the qualitative data, and that will address your RQ completely. So connect the qualitative observations to reason the quantitative data in your analysis, in your conclusion. According to precision and accuracy, I think most of the students are aware of it. This is given in the random errors or the error analysis every time by the teacher. I will just skip this slide so that we can get along well. So here are the three graphs given when we do the data processing. And everyone can very well say that which, I'm not going to the voting one. Everyone is very clear about, I think, which one is a more complete and more relevant graph. Which one is that one? The third one, where you have the standard deviation bars with every observation, the R square value, both the axis named with the units given, right? What is the difference now? What is the difference in the second graph? Why it is not the best one? It just shows the scatter with a standard trend line drawn. If you see the second graph and compare it with the third one, in the second graph, we see many data points are very far off. Hardly two of them are lying on the trend line. But when you come to the third graph, you see because the standard deviation bars are there, the scope of your data increases. There is the variation given that though you have recorded that data point, what is the variation? It can still lie on the trend line. So it gives a better measure of correlation between the two variables. It helps you for your analysis and evaluation. So that's why the third one is the better one or the excellent one. So it enables you to observe the patterns, trends of the whole data set, address variability, uncertainty, anomalies. Standard deviation also reveals that your data is reliable, though they are not lying on the trend line actually. Here, we move to now the analysis escalator, how you can improve your analysis. So you see here, the mean rate, there are two types of analysis statements given. The mean rate of oxygen production increased from trial 1 to 3. Of course, it's very much evident from the first graph. Value is given at 40 degrees C. If you check the second graph, second statement, it gives an analysis. Transpiration increased 2.6 fold. After this it explains the whole graph. So anyone can say that analysis B is a better one. So you see how you can enhance your analysis, how you can climb the stairs. So you state that trend. When you say that the graph is increasing up to 40 degrees centigrade and then the effect is decreasing, it is just an observation. You are stating the trend. Now you will recognize that what is the biological cause behind it. And more enhanced, you reach the final culmination when you say how this relates to your RQ. How it relates, it proves, it disproves your hypothesis. So this is how you can improvise your analysis. Analysis is just not describing what is given in the graph. It is relating it to a biological context or giving a cause why this is happening. Here is again a challenge. The rate doubled 0.5% and 1% substrate concentration, and this doubling indicates that the enzyme molecules had more frequent substrate collisions. If the concentration increases, the number of substrate molecules or particles are increasing, so there is more collisions. So that is a better statement. You also know the biological mechanism which is enabling it. Here we are concluding. So this is the conclusion. Remember always, it's very important and very simple addition to your IA. In the conclusion, begin with your RQ. Analyze your hypothesis, whether your hypothesis is proved to be appropriate or it is disproved with your investigation. Write something about the concrete about the validity of your investigation and the data collected, and also conclude with the biological concept which your research question has answered. Don't go vague over everything what you have done. And finally, these are the 6 essential points which can be your key takeaways from today's webinar. Choose a focused, personally relevant RQ. Design a controlled, the control is very important, repeatable methodology. Collect sufficient quantitative data with replicates, with trials. Use appropriate graphs and statistical analysis. Okay, it was not possible for me to include the statistical analysis in this 1 hour webinar, but IB in its teacher resource material has very well given a chart for the statistical analysis, and the teachers can follow that chart, helping the students with statistical analysis they need to do. Because the choice of statistical analysis, that guidance, if the teachers can give to the students, it will be very helpful, because the students get confused which statistical analysis to be done. But that chart gives a very clear indication. And then for the statistical analysis, you can find any online platform. Most of the online platforms have statistical tools. You have to just include or upload your data and they give you the test which you want. Evaluate limitations and their impact on conclusions. It's a 100% must. You include what are the strengths of your investigation at the end in the evaluation, the limitations, giving limitations, identifying limitations, recognizing that these were limiting your study is a very many students, like, they are not very comfortable saying the limitations or presenting the limitations of their study. But you are students. IB expects you not to be perfect. So if you can recognize the limitations of your study, this further gives a clear indication that you have developed that scientific aptitude, and the next time you will do that experiment, you will surely try to improve your experiment. So give the limitations with corresponding improvements. Make the improvements reliable. Don't say that for this I will do a PCR or a very sophisticated test. Give very simple methods, like instead of using a glass pipette where I was having difficulty for pipetting, I will use a digital micropipette. Give doable things which are very simple. Structure your report clearly using the IA criteria. That's it. So now the webinar is open for questions. Yes, please. I hope I have, I tried to complete the most of it.
Ma'am, I think you did a very good job. We were all on our toes. Anybody has any questions? No, I think questions they asked between the IEs, between the webinar itself. Yes. Okay, I have pasted a link for feedback from you guys. Please go through that. So I'll do the closing now. Thank you so much, Dr. Sarkar. That was such a fantastic and fun session. I loved how interactive you made everything. Thank you for keeping us on our toes and finding so many creative ways to get everyone answering and involved. Big thank you to all our audience too. Your energy and participation really brought this webinar to life. I hope you all leave today feeling confident and ready to tackle those biology IAs. I would really love if you can give feedback on chat or on Padlet. Actually, both would be better. So we would know, and we would and encourage us to organize more such webinars.
Thank you, Ms. Deepika and Mr. Aniket for hosting this webinar. It was a wonderful experience interacting with peer educators and the students. The students were very quiet. I hope with the teachers along with them, they are little concerned, conscious.
Probably,
But
yeah.
yeah, we will see again.
Yeah, you were a real fine, Deepa ma'am. Thank you so much.
Thank you. Thank you so much.
Bye, guys.
We will connect with each other, Ms. Deepika.
Definitely.
Thank you.
Thank you. Bye.
Bye.
Thank you, ma'am. Thank you, guys.
Thank you.
Bye.
Thank you. Miss Deepa, can I connect with you on LinkedIn as well?
Yeah, sure. Nidhi.
Yeah, thank you.
So thank you, Deepika ma'am and Deepa ma'am for the wonderful session. I thoroughly enjoyed it and hope connecting with you all on LinkedIn also.
Thank you.
Definitely, ma'am. Thank you so much.
Okay.
Thank you. I will leave. Thank you, Aniket.